My name is Sébastien Mosser. I was first passionated by the neuroscience and studied deeply about Alzheimer's disease, which I did my PhD in Lausanne at the École Polytechnique Fédérale de Lausanne. And then I was really moved toward the need to go for entrepreneurship, really very interested by startups and by the dynamics it means, and what brought me after a different step to Neurix. I'm a neuroscientist, so very interested into all neuroscience, and the aim and disappointment is always to have to work on mice to produce data that will be applied to humans. So here I think the great work and great potential for induced pluripotent stem cells and human embryonic stem cells. The problem of Neurix and the problem in general of industry is that data are first and drugs are validated on mice before going to clinical trials. But a mice is very different to humans, pharmacologically speaking. So it means that 90% of this drug will fail on human being tested, which make a very high cost, wasted money for industry, and there is a clear need for better preclinical models. We develop in Neurix, thanks to human embryonic stem cells and human-induced pluripotent stem cells, what we call the Minibrain, which are three-dimensional neural tissue derived from embryonic stem cells. And with this aim, we want to mimic closely the human brain and increase the selection and the right selection of the drug for pharma industries. So I think the main challenge we have here is having an organoid or a mini brain into academic scale is quite easy and can be developed. But our aim and our main challenge is really to upscale, to be compatible with high throughput and with special demands and research projects of our customers. First, they have to be alive, very simply. But more importantly, they can be alive and start differentiate, which will be a big drama because we cannot use them later for different procedures. And then we have to make sure that they grow enough. As I told you, we're currently into industrialization, so we have to make sure that we have enough cells in order to decrease our cost and to be competitive on the market. I think stem cell is one of the biggest perspectives in terms of medicine, because it could be, in Neurix, we essentially focus on drug development. We could create like human-based organoids to after select the right drug candidate and study some human-specific mechanisms. But in addition, regenerative medicine and creating tissues from cells directly taken into the patient is just a great opportunity to solve challenges that have not been resolved today. We use it at the very beginning, which is the more crucial part of our process. It's culturing the human embryonic stem cells or human-induced pluripotent stem cells so that they keep a very high quality standard. And thanks to the high quality standard, we were then able to have a clear and reproductive differentiation to generate our mini brain and satisfy our customers. I think I'll just test it. That's the most important step to be done. But of course, to have the right controls. So first, changing the culture media is a very high stress for cells. So this has to be done really on a clear basis upon dilution with the previous culture media, the first step. And second, of course, a very rigorous quality control has to be done. I guess your customers do after differentiate these embryonic stem cells or iPS cells. So they should check that at the end, essentially either via qPCR or doing some gene sequencing to make sure there is no modification, and indeed make sure that they obtain the same product. We clearly recommend the NutriStem because we can keep our high quality standards, we can industrialize our techniques thanks to this, due to the quality but also due to the cost. So we chose NutriStem essentially based on a cost and quality ratio, and this is something we highly recommend either to academic laboratories or to industries that will to pursue into this direction. So Neurix starts from embryonic stem cells, and we differentiate these cells toward neural lineage into a three-dimensional tissue, three-dimensional organoids, which we call the Minibrain. This human Minibrain can be used either for neurotoxicity screening, even a high-throughput compatible setup, but also we're able to do some very custom projects. For example, we have some customers that want to check about the virus spreading into the tissue. We check about virus spreading, which cell type does it infect. We have some others that want to check about the glioblastoma invasion into our tissues, and are of course welcome to other research projects that will imply very specific mechanisms. I think organoid is the future of biology, because very simply, take whatever organs you take, even the skin that appears to be flat, it never be cultured in 2D upon a plastic surface. So the best way to reproduce human features is to culture them into a natural environment, which means a 3D environment. And at the moment, these techniques can be difficult to apply because of technical issues. This is a new field, but I'm sure that in the next 5 years, upon mastering of the different organic cultures, this will be a great field. And I think great opportunity to work with human-specific organism and environments. Well, this is a question that is partly confidential. I may not be able to share all our procedures, but all the expertise of Neurix has been built since 15 years on the expertise of Professor Krause lab, which is one of founder, and Professor Stopini lab, which is also a co-founder of Neurix. This has been 15 years of optimization to have organoids that show similar features to humans, that are reproducible, and that share the highest quality for our customers.